Welcome to week 12 office hours, AP Biology students. And this time we will go and discuss cell communications, the main steps. This is AP Biology 4.1 through 4.4 topics. Cell communication is super important, not only amongst the cells, but within an organism for multicellular organisms, but it's also important for single cell organisms, eukaryotic or prokaryotic, doesn't matter, to know about each other and the intentions and the actions. So communication is important for the cells too. Now, so the point in here is not really knowing the itsy bitsy tiny details of every single steps, like how the epinephrine is acting through a receptor and the cascade inside, how many layers we have. For the important aspect in here is to understand the main steps in cell communications, to see how the ligands, depending on whether they are hydrophobic or hydrophilic, they act differently, and the receptors can be different, and the receptors on the cells, whether the cell has a specific receptor, depending on which cell in an organism is responding for the signal, for the ligand. Not all of the cells contain the same receptors on themselves. So, for example, the T cell in the immune system reacts differently to an antigen-presenting cell compared to my red blood cell. The red blood cell doesn't have that receptor, so it will not be able to respond to a certain ligand or certain message. Also, it's important if something is changing in that cascade in the communication, like the ligand, the communicating molecule, or the receptor, or the transduction proteins, the response will change. That can lead to cancer, or that can lead a different response than from a normal response. But before that, getting into the details, let's go over and check the ligands. That's first one. Ligands are the molecules that the cells use for communications. They can be hydrophilic or hydrophobic. Depending on whether they are philic or phobic, they can enter the cell or not. Hydrophilic is, for example, proteins, and they cannot enter the cell. They need a receptor embedded in the membrane. The hydrophobic is kind of the lipids. One of them is the steroids. They can pass the cell membrane, and they have their receptors in the cytoplasm. Let's draw a cell in here and get some of the markers out. So we have inside, in the cytoplasm, there is a intracellular receptor. The steroid will be, for example, a circle, so the circle can pass the cell membrane, and the ligand forms a receptor ligand complex inside of the cell. we have the nucleus in here. The nucleus contains the DNA. And this receptor ligand complex can enter into the nucleus, and it will act as a transcription factor. Transcription factor, gene expression, and it goes out. We will learn about that later, how genes can get expressed, and it will make a protein for inside of the cell or for exporting that. We have that the proteins, on the other hand, those ligands, or we can say it hydrophilic or ions, anything like that, they have a transmembrane receptors. It can be the transmembrane receptor like, here is the receptor. This is the ion. Okay, ion channel. And this is receptor mediated ion channel. So we have a triangle that is going to sit here into the receptors site, and it's going to make a conformational change in the receptor and will let the ions go into the cell. So we are talking about now the receptors. It can be intracellular, and also it can be transmembrane. The second one is, this is one of them. The second one, they're not necessarily on the same cell, but it's a receptor, transmembrane receptor, attached to a G protein coupled receptor, because it's coupled with the G protein. And also we have a enzyme in here, cyclase, and it's making cyclic AMP out of ATP, adenylyl cyclase. This G protein, when something is going to get attached to here, this is the ligand, the sphere will get attached to, it will activate the G protein. The G protein moves over to the adenylyl cyclase to activate that enzyme to make cyclic AMP, and cyclic AMP is getting into a cascade mechanism, acts as a secondary messenger, and activates other molecules, enzymes, as well as amplifies the signal. The third one is going to be, I put it in here at the bottom. It's something like this. This will be also a square. Oops, not this one. Like this. And tyrosines are here. These are amino acid, the tyrosine. And when they, let's do that. It's a not a this, but a rectangle shape. Okay, for making it different a bit. And this is the ligand for those, and they can work only in twos. Dimer formation happens when they bind the tyrosine kinase receptor. Tyrosine kinase. So this is an enzyme in here with the receptor combined with, and they will get activated when the ligand binds to the receptor and they start to activate with ATP, so they will have a phosphate group attached to all of those binding sites, and they will activate proteins on their ways for the transduction. What else? So the receptors, only cells with receptors can respond to the signal. If the cell does not have the receptor for that specific ligand, that cell will not respond to the signal. This is the receptors or reception. The next step will be transduction. Transduction is a cascade mechanism, like enzymes or activating other enzymes or blocking other enzymes. It can be done by phosphorylation. Enzyme named kinase. There are many, many different kinases. So phosphorylation to usually activate, but not all the time, activate an enzyme, or dephosphorylation. Dephosphorylation. Dephosphatase is taking away the phosphate group. Now, in the transduction process, we can just also draw it like X molecule or X substance is going to activate a Y substance, and it's going to activate a Z substance. This is a cascade. Now, arrow means activating, but if I draw here a line with a flat top on it or perpendicular to it, like I will put here a letter P. So for saying this is another substance, it will inhibit the Y to turn in Z. So X activates the Y would activate Z, but the P does not let the Y to activate the Z because of this. So if we have another blocking in here, like M blocks P, and if this is activated, blocks, so it's not going to work, so Z is going to be produced. But if this one is present, but M is blocked, nothing is blocking P, so it's going to block the transduction from Y to Z, so it's not going to happen. So these are the symbols what I would like you guys to embrace. Now, the last one is going to be the response after the cascade. So reception, transduction, and response. The response can be depending on the different functions and the enzymes over there. It can be cell growth. It can be cell division. It can be secretion of a molecule, like in here. It can happen like enzyme activation, enzyme blocking, and also it can happen apoptosis. Apoptosis, the cell decides or gets the message that the cell needs to eliminate itself from that big mass. Now, you would think about that this is very bad, cruel, but think about the formation of the fingers. We have, as embryos, limb buds, and in order for the fingers to form, the buds, in some places in the bud, the cells got to kill themselves. They have to go through apoptosis to separate those parts in the buds to form fingers. Okay, now, how to tie that to evolution, or what we learned about that in here. Evolution. Many organisms, many cells have the similar receptors all around in different species. They have transduction cascades very similar, responses similar. So this really kind of shows a unity among the species that really supports the evolution, that we came from one. The last thing what I would like to talk about is the type of communications. There is the short distance communication and the long distance communication. The short distance communication can be autocrine communication. Autocrine, that means the cell produces a growth hormone and will have a receptor for that growth hormone, so it induces itself to grow like cancer cells. So this is the autocrine. The other one is the justacrine. Justa means really touching each other, so they have to have contact. This is the immune system, T cells, as the antigen presenting cell combines with the T cell to activate the T cell. The another one is the plasmodesmata in the plants. The third type is the paracrine communication in close vicinity. They are not touching each other, the cells, like neurotransmitters from one neuron to another, or from a receptor to the neuron. The long distance communication is the endocrine communication, endocrine, based on the hormones, growth hormone, thyroid hormone, growth hormones, parathyroid hormone, testosterone, estrogen, insulin, glucagon. So those are all endocrine stuff. Lastly, so if there's a change in the ligand, or change in the receptors, or change in the transduction, that means, just a sec, that means these changes are affecting the responses, and the response will be different for that. Change in any of the steps can change the response, and this is where sickness will kick in, or the abnormal response will happen. That's all basically for today. Have a good one.